Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about apoptosis or programmed cell death. What is programmed cell death? It is the genetically controlled death of the cell, which is otherwise called as apoptosis. It is different from normal necrosis in this way that necrosis is energy independent, but apoptosis is energy dependent and a controlled process, genetically controlled process. For example, apoptosis takes place during the finger development of an embryo. Okay, so the main enzyme which is involved in apoptosis is caspase. Caspase, the full form of caspase is cysteine dependent asparatate specific protease. It is the main enzyme involved in apoptosis. The apoptosis takes place in three different pathways. One is extrinsic pathway, second one is intrinsic pathway, third one is granzyme mediated pathway. So the first one, extrinsic pathway, where the programmed cell death or apoptosis takes place due to the presence of a death receptor on the cell of the target cell. One ligand will attach on this death receptor. The association or interaction of this receptor phase and the ligand phase L will activate another domain, which is FADD. FADD means phase associated protein with death domain. Okay, so this activation recruit or form a death inducing signaling complex, which is called as DISC, death inducing signaling complex. Okay, so DISC then activate caspase 8, and in turn caspase 8 activate caspase 3, and caspase 3 next leads to the exclusion pathway or other downstream processing, which leads to the death of the cell. Next one, intrinsic pathway or otherwise known as mitochondrial mediated pathway, because the main role in intrinsic pathway is played by the mitochondria. It releases cytochrome C. Cytochrome C then form the apoptosome, which leads to caspase 9 activation, and finally to caspase 3 activation, and which leads to the programmed cell death or apoptosis of the target cell. Next one is granzyme mediated pathway. So this granzyme pathway is mainly used by our cytotoxic T cell and NK cell or natural killer cell to kill virus infected cell or tumorous cell. So these cells secrete some substance, porphyrin, to create pore in the target cell, and through this pore the granzyme A or B, mainly granzyme B, enter inside the cell. Okay, then after entering, the granzyme activates the caspase 10, and in turn caspase 10 activates the caspase 3. Then caspase 3 leads to the death of the cell. Another thing, this intrinsic pathway is mainly regulated by BCL2 family of protein. Okay, so this BCL2 family of proteins mainly included anti-apoptotic member and pro-apoptotic member. So pro-apoptotic member, which promotes apoptosis, and anti-apoptotic member, which inhibits apoptosis. So anti-apoptotic member includes BCL2 or BCLXL. Okay, and pro-apoptotic member is BAX, BAD and BIK. So you have to remember these family members or these family member proteins. Okay. So it is all about apoptosis or programmed cell death. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.